see some people are still joining in, but it's not be late. It's my pleasure today to have Dr. Carlos Chaccour. Is a Venezuelan researcher currently based in Spain. He's a trained internist with clinical experience in remote indigenous communities and in the Venezuelan Amazon or in Mozambique as well. He obtained his PhD in 2015 working on slow release Ivermectin formulations, and his research now focuses on the development of mosquito killing drugs as complementary vector control strategies for malaria control. He's the chief scientific officer of the unit-funded BOHEMIA, and it seeks to propose ivermectin as an additional tool to reduce malaria transmission. So thank you, Carlos. I just remind you, for those who have never done this, that you can put all your questions in the chat while Carlos is speaking, and I will answer them at the end. He'll answer them. Okay, thank you, Carlos. You can now share your screen.
Okay. Just 1 second. Can you see as well?
Yep.
All right. So thank you, Maureen. Also, Maureen works partly for BOHEMIA as well. She's the super duper postdoc at Kenya doing all our mosquito processing, etc. Just too humble to say it. All right. So Maureen asked me to talk about something, and of course I will talk about ivermectin because some people would say I don't talk about anything else. And I'm going to talk about a bit of history, how ivermectin came to be and in which context and why it took off so high and so quickly and so importantly. One slide on mechanism of action and spectrum, the impact of on NTDs, and then malaria, of course, and I'm going to spend maybe a few more minutes there because it's my field. A few slides on recent controversies regarding ivermectin and COVID, which has been led to an enormous amount of trolls and hate. And then we can have a chat about where are we going, etc. So Marina and I were talking about this, and she didn't know this statue. I think this one is a copy, but the original one, as far as I know, is in front of the main building in WHO in Geneva. And this is a statue that commemorates the success of the onchocerciasis control program in West Africa. And what you see here is a young man, maybe in his 40s, blind and being guided by a boy who is not blind yet, but probably will be blind because in the 50s and 60s, onchocerciasis was really the queen of West Africa. In all the Volta Bay Basin, this disease had led to so many people blind that there
Carlos? Carlos, can you hear us? Because we can't hear you. Sorry, guys.
Okay, now better?
Yep. Yeah. We can hear you at all.
So my sound card has an issue with my computer. Okay, so onco is a disease caused by filari and transmitted by black flies. And these black flies breed in areas with water that has high levels of oxygen. So in villages that are close to rapids or wild water, they are prone to have a massive amount of transmission of onco. And that created hyperendemicity, and that led to people getting blind in the 40s. So this was whole areas with phantom or ghost villages, half abandoned, because most adults were basically blind or half blind, and children were guiding them. But these children would also start losing vision in their 20s. So this was a huge problem. And then Robert McNamara had been Kennedy's Secretary of State, had handled big issues such as the missile crisis in Cuba, and then he went on to be in the World Bank. So somebody in 1972 took Robert McNamara for a trip into these areas of West Africa, and he had the opportunity to witness first person the scourge that this was causing. And given that he was in his new World Bank position, this allowed him to help these communities as much as possible. So together with the UN and WHO, a program was started, which is the Onchocerciasis Control Program in West Africa. And here on the right is Robert McNamara in a visit to Ghana in 1973, I think. So this program consisted in areas of the Volta Basin, basically, although there's some of extreme West Africa here. They would spray areas of the river that served as breeding sites for black fly larvae, sometimes with helicopters, and they would rotate insecticides, expand the area of impact. If you wanted to control in a little bit area here, then you would expand and create a buffer area around it. They rotated the insecticides, they did strong M&E, they had a strong community engagement and a lot of political buy into it. And the program was a success, and there's the statue that I showed you. But even then, it was seen that it was not enough. And then came this game changer, which is ivermectin. Ivermectin is a drug that basically binds to some channels in the neural junction of invertebrates and keeps the channel open, and this causes paralysis both in helminths and also in insects. So it has a huge spectrum, including several arthropods, but also the filari that cause onchocerciasis. And the story is more or less like this. Developed in the 70s, it was first isolated in the dirt of a golf course in Japan, and developed in a joint program from the Kitasato Institute in Japan and Merck in the US. And this is the first publications from 1978, Satoshi Omura talking about the new molecule and the efficacy of it, talked by Campbell. This is antimicrobial agents and chemotherapy in 1978. This is a patent 1 year before, granted to Merck, and it became a hit. It was the #1 sold antiparasitic for veterinary use worldwide. Around $1 billion is the annual market. Now it's pretty much more. And as early as 1981, they saw that Onchocerca cervicalis, which is the Onchocerca of the horse, disappeared in areas where they used. Wait a second. Can you hear me now?
Yeah.
Yeah, okay. I just fixed something. All right, so the Onchocerca of the horse disappeared in areas where ivermectin was used for veterinary purposes. So already in 1982, they say, hey, we have this huge program in West Africa controlling Onchocerca. Could it work for humans? And as early as 1982, there came this famous Assis paper in the Lancet showing that ivermectin basically doesn't kill, but sterilizes adult filari and prevents the clinical consequences of the disease. It doesn't cure you, but it prevents you from going blind. So then Merck did something unexpected, and I'm not sure it has happened often afterwards. And in 1987, they created the methylxanthonation program. This was Roy Vagelos was the CEO of Merck back then, and he decided that the company would donate as much ivermectin as needed to as many people who needed it for as long as it was needed until Onchocerca was eradicated. By 1998, the efficacy of ivermectin against other filaria, LF, was well established, and it was added to the donation program. And this is old, but by now 2015, they had reached 2.7 billion. Right now, it's probably more than 3 billion doses that have been given for free. And in 2015, also, Omura in the center and Campbell shared the Nobel Prize with UU2, and this was a whole Nobel Prize given to antiparasitic drugs. These two guys for ivermectin, UU2 for artemisinins. So basically, recognizing how impactful this drug had been. Talking about impact, this is the number of doses that have been given from the late 80s until today. And you can see that we're reaching about 400 million treatments yearly. So this is 400 people that receive treatment every year for the last few years. And you can see it's onco, it's LF, it's both. And now you can see triple therapy, and I'll talk about that later. So what does that mean beyond number of treatments? And what this means is that in these areas where the disease is endemic, the number of people that do not require any more treatment because the disease has basically been reduced to a non-public health problem or eradicated or eliminated from those areas is reaching 180 million people. 180 million people protected from losing their eyesight or from having elephantiasis. This is a lot. This is a huge impact. Recent game changers, I mentioned triple therapy, and just as it happens with onco, with LF, ivermectin does not kill the adult worm. So the adult worm can remain alive in the under the skin, in nodules, or in the lymphatic vessels, and it gets sterilized for a few months after each dose, but it doesn't get killed. So this forces us to repeat mass drug administration yearly for a long time until transmission is so low that there are no new cases appear. But 2 years ago, there came this publication, although the news have been out there since 2015 at least, in which a combination of albendazole, ivermectin, and DEC, all three together, basically sterilized the adult worm forever, i.e. killed it. And what you can see here is how they gave randomized people into either the 3-drug regimen or 2-drug regimen once, or 2-drug regimens once a year for 3 years, which is the standard of care. And what you see is the number of microfilaria in the skin or in the plasma. And you see that in either of the current regimes, if you give it yearly, you can manage to keep a low microfilaria burden. If you give it only once in 3 years, you don't get that. But if you give it only once and don't repeat it ever again, you basically sterilize the whole thing. So this means a single dose given to all these areas could help us save millions of doses in the future. So this is a game changer. Other problem with the ivermectin is loa loa. And loa loa, it's a relative, well, it's an ugly parasite, but it doesn't kill you. And this parasite wiggles under your sclera, and it's a horrible thing, but it doesn't cause many more clinical symptoms beyond some swellings and mild disease. However, if you have a high LOA burden in your body, you take ivermectin, apparently ivermectin is so good at killing it and so fast that it can cause a severe encephalitic reaction, and people have died after being treated with ivermectin if they have had a high burden of LOA in their bodies. So that prevents ivermectin from being used in all of these areas where LOA is highly endemic. So basically all of Gabon, you cannot use it. Equatorial Guinea, you cannot use it. Most of Cameroon, huge areas of Central African republics, huge areas of DRC Congo, all the south of Congo. So in all these areas, you couldn't use ivermectin. So the containment of onco-NLF was under threat. And then came smart people and they came up with this idea of this quantitative technique that gives you results in real time. And you basically use a smartphone, you put it on this box, you have a square capillary, you take blood from people in the area. The risk of this encephalitic reaction was only described about 15,000 more or less, originally even 30,000. So basically, if you have less than that, ivermectin could be used safely. So they use this smart technology to not really count the parasite, but count the wiggling of the red cells. So you put the sample here, and in 1 minute, and there's videos of this in New England Journal of Medicine, I invite you to check them out, it gives you an either red, yellow, or green label in here. Basically, if your parasite burden is low, then you can give ivermectin. If the parasite burden originally was 20, later on they reduced it to 15,000 because they had some more severe reactions, about 15. It gives you yellow, says you can give it, but tell people they may not feel so well. And above 15 or 20,000, please don't give it because you risk killing them. Right? So this is great because this allows you to treat everyone who needs treatment and is not at risk of severe disease. This reduces LOA itself, and basically in not much time, these areas could be receiving ivermectin because you're effectively avoiding these side effects in people who have high risk. Another game changer is the indication of ivermectin for scabies, and it's been accepted in France, in Australia as well, who have the huge problem. And this is from 2015, this work by Romani and the Australian crowd, basically showing that if you give this in mass drug administration, 2 doses, the prevalence of scabies in the Fiji where they did the trial was 32%. It's a huge prevalence of scabies. But they managed to reuse it with ivermectin in 94% reduction of scabies prevalence after a year. Compare that to just 62% reduction with permethrin, or half of that with the standard of care. So ivermectin has also walked with a very firm foot in the scabies area. And now a bit on my side, ivermectin does kill mosquitoes. And we've known that since very early. In 1979, the first paper showing that ivermectin killed ticks was out. In 1885, the first mosquito experiment was done by the Gruppo Pampiglione in Italy. From 1989 to 2009, so 20 years, this was basically a paper every 3 years. So some people took interest in this. There was an early paper of some guy who took ivermectin on himself and then fed hundreds of mosquitoes on himself and showed that it reduced N equal 1. There was a field experiment done in Papua New Guinea, and not much more. And then in 2010, there came 11 papers in 3 years in this period, 2010 to 2014. So ivermectin for malaria started to take off. And basically because by then it was very clear that we had a problem with what we used to call back then outdoor biting. So mosquitoes that bite in places that are not protected by IRS or nets. Now we call that residual transmission, and we have broadened this to include mosquitoes that bite dusk or down, mosquitoes that feed on animals, mosquitoes that feed inside the house but then exit the house early. So having a drug that is in the body and basically kills mosquitoes that feed outdoor or indoors or everywhere is an interesting complementary strategy. And now we have tons of trials and tons of publications, and I've even stopped counting them. So what's the evidence? Insectary, observational, and randomized trials. So in the insectary, there's been more than 30 studies, and it's difficult because they use different species, they give ivermectin in different ways, they follow mosquitoes for different times, they use humans, they use animals, but what is clear is that if you feed mosquitoes on humans that have taken the drug or animals that have taken the drug, they die at a faster rate. This is my work from 2008, published in 2010, and this is the mortality rate of control mosquitoes fed on non-treated volunteers, and this is the mortality rate of mosquitoes fed on treated volunteers. This accelerated mortality in controls is what happens when you put an MD to do entomology work. Normally, this would be like here, right? And the gap would be much bigger. This is my fault. If you repeat this experiment after 2 weeks of taking the drug, then there's no difference. So there's clearly a dose-dependent effect there. That is, the more ivermectin that is in your body, the faster and in more proportion the mosquitoes will die. That's from the insectary. And as I said, that's just one. There's 35 other papers. I think observations in the field, I really like the work Brian Foy has done. I think something very smart to do. What you do is basically you take advantage of this mass drug administration with ivermectin that happens in West Africa, now in many other places, but mostly in West Africa. And you go and collect mosquitoes before, during, and after. So it's not a clinical trial because people are getting the drug for other reasons, but you can do your mosquito collections and still see the effect of the drug. This saves you a lot of trouble, regulatory-wise, money-wise as well. So basically, Brian and his team have collected mosquitoes in several sites in West Africa, and they have shown, that the survival rate, here you see controls in blue, ivermectin in red, and this is villages and mosquito collections, and they follow mosquitoes that they have captured before for a few days in the insectary and see how long they live. And the survival rate basically drops sharply at the day of MDA, and this lasts for about 10 days. And in the controls, there's no change. That's one thing. The other thing that happens is that the mosquitoes that you capture not just survive less, but they are much younger. So the age structure of the mosquito population is shifted to much younger ages, which are, as you know, less able to transmit malaria. And it doesn't happen in the controls. So this drug is working in the field. And finally, the sporozoite rate, which is the proportion of mosquitoes that have parasites in their salivary glands, it is, in this case, inverted. So it remained high in controls, and it went quite lower in ivermectin-treated areas. And that lasted for about 2 weeks. So this is disruption of transmission. The question is, oh, very nice, Carlos, but then how come all these areas receiving ivermectin for all these years still have malaria? Well, because a single dose once a year is probably unlikely to reach a threshold that can have epidemiological consequences, and we can measure it. And finally, this is also from Brian, and this is the Rindemal trial. They randomized clusters of communities, 2 groups. One group received a single dose of ivermectin, and the other group received 6 doses of ivermectin every 3 weeks. And they saw a 20% reduction in malaria incidence in children that live in the intervention villages. So this was the first evidence that this could have impact in the field on a randomized manner. So how does it work? And I like this cube. I think it's very informative to show that the efficacy of ivermectin for malaria would work along three axes. One is how high the concentration goes. Basically, the higher the dose you take, the higher the concentration that you will have in your blood whenever they bite. And as I said, the higher it is, up to a certain point, you will see more mortality and faster mortality in those mosquitoes. Second is duration. It's not the same to have a curve in your plasma that is prolonged in time and mosquitoes are dying and biting on people who have these levels for a long time, that just a peak that lasts a few hours. So these two combined are an area. So this is levels and this is duration, and area under the curve is what you see here. But then the third dimension is brought by how many blood sources are treated. If you, the more humans you treat, the higher the impact you would expect, because there's less clean blood, to talk in Harry Potter terminology, to feed. So mosquitoes would have a problem. And if you include livestock there, and those mosquitoes that like biting on cattle and then coming back to biting humans, then they have a huge problem. So those are the three axes, and that's part of what we're testing in Bohemia, together with Maureen, and we will be working in Tanzania and Mozambique. We're delayed a year due to COVID, by the way. And the One Health concept, I'm just going to throw a few ideas there because I like the idea, and this is, I think, one of the most unique things we do in Bohemia. We're taking into account that third axis of blood sources. So treating or including the One Health concept allows you to get very creative, and you can use long-lasting formulations. You can consider other molecules that you wouldn't consider on humans. You also have to have other problems, like if you treat a cow, how long do you have to wait until you can milk it or slaughter it? So where would this be useful? This is malaria across Africa. And this is the same information, just in percentile. So basically, we took this, we made it 100, and then we made a map based on that. And this is cattle. Again, the same thing, the true data, and then percentilized. And this is Anopheles arabiensis, the Bruce Willis of mosquitoes. This species likes to feed on cattle as much as it likes to feed on human. It likes to feed outdoors. It likes to feed early in the afternoon or early in the night. So it's a problematic species. So if you combine these three maps, you get this. This is areas where there's a lot of malaria, a lot of ton, and a lot of arabiensis. So basically here, and also here, sometime here, it's areas that probably would benefit a lot from this strategy. And we took that to the district level, and it looks quite nice. And you see Bohemia is working here and here, and there's a lot of work happening right here in Burkina. So another thing you can take advantage of having a non-human target is that you can do this kind of. This was my PhD thesis, and we basically did an implant of silicon that has two rods, and this rod elutes ivermectin slowly. This is pure silicon. And this is one of the last experiments. We tested it originally. My thesis was in rabbits and pigs. And this is something we did in Tanzania. We took 3 calves, and 1 was controlled, 2 received implants. And then we took blood on day 0 and fed mosquitoes on them, and took a sample, kept it to determine the levels of ivermectin, and repeated that every 2 weeks for 44 weeks. So what happened is that the ivermectin levels in both treated and untreated, in both treated calves, well, they peaked at around 2 weeks and more or less stable for 16 weeks, and then after 6 months, they dropped below 5 nanograms milliliter, which is a concentration known to kill a large percentage of mosquitoes. But you can see detectable ivermectin for 40 weeks. So what happened before implantation? This is survival of control mosquitoes feeding on control calf. Basically 50% survive 11 days. And in the treated ones, well, even more, right? So no difference here. 2 weeks after implantation, this is the control. And we basically smashed the dose feeding on ivermectin. How about 12 weeks? Here's the controls, and here's ivermectin. How about 24 weeks? Again, a gap there. How about 36 weeks? Again, a gap. Although you see that dose response is reducing here. So there's many ways to look at this. Mean survival, it's, well, you see, right, in the controls, they more or less survived between 6 and 9 days in a mean along the whole experiment, right? In all these experiments that are 2, 4, 6, 10. And in ivermectin, you basically had a huge gap. They survived much less for a long time until ivermectin dropped below 6 nanograms milliliter. This is ivermectin, and you can see this scale. 10-day survival, which I think is very interesting because these mosquitoes will not become infectious, was varying, and I think this shows the weather. This was done in a biosphere in Ifacara. But if you look at the ivermectin ones, basically none of them survived 10 days for the first 28 weeks of experiment. And then you model this, model tells you that you can have a huge impact on malaria incidence or prevalence, clinical incidence or prevalence if you treat only cattle, depending on what's the proportion of cattle, of mosquitoes that are feeding on that cattle. Another interesting point is that if you include livestock in your equation, these animals also have parasites. And these parasites in their intestines basically eat their nutrients, which basically preclude humans from the economic gain and food security of this. If this cow gains less weight or produce less milk because of parasites, then this human also has less resources. So if you treat them with ivermectin, you don't just prevent mosquitoes that feed on this cow to feed on this human later, but you make the cow grow more and produce more milk, and this person has more money to invest into healthcare. So the synergies with NTDs and malaria, this is LOA you showed before, and this is oncofoci, and you can see the huge overlap with malaria. So if this was a strategy to be drawn out across Africa, basically all these areas would benefit from malaria prevention, if we prove it's effective. It's not proven yet, and from onco prevention, which is already proven. So finally, maybe 5 minutes on COVID controversies. So I've been doing my homework on ivermectin for 12 years. And yes, we know it inhibits viral replication of many viruses, including flaviviruses, yellow fever, dengue, chikungunya. It inhibits the virus of the porcine flu, porcine lung fever. It inhibits a myriad of all other weird viruses, the Similiki virus, huge other viruses, many other viruses. So when the pandemic came up, we thought, well, let's do a trial of ivermectin because, I mean, this is a disease with no treatment, and this is a drug that is very safe, so let's try it. We were planning for a trial, and then came out Kali et al. Kali et al. Is a paper that came out of Kili Wagstaff lab in Australia, in Monash. And basically, they incubated SARS-CoV-2 in Vero cells, and they added ivermectin to this. And what they saw is that at 2.5 micromolar, the replication of the virus is reduced in half. And at 5 micromolar, the replication of the virus is basically 99% reduced. So this is a huge impact, and everybody was very excited. But then you have to ask me, okay, how much is 2.5 micromolar cost? And then I'll tell you, 2.5 micromolar is about 2,400 nanograms milliliter. Now, 2,400 or even 800 nanograms milliliter is between 50 and 100 times the maximum concentration that you get in your blood after you give a standard dose that you will use for onchocerciasis. So yes, this is very exciting, but then these concentrations cannot be achieved in vivo, at least not using safe doses. You would have to eat, I don't know, a block of ivermectin like a cheese. That's not safe. But does this mean that we should not try it? Well, I also disagree with that. I think we should test it because ivermectin, as I said, I've been doing my homework on this molecule for a few years, have other effects. It acts, it immunomodulates your immune response. It reduces the amount of IL-1 and IL-6 one produces in response to infections. It can interact with nicotinic acetylcholine receptors. It has active metabolites. It can accumulate in the lungs. So there are reasons to test that even if there is a PK IC50 mismatch. But what there is no grounds to do is to make it a national policy. I'll get to that. Because there's drug-drug interactions. If you take ritonavir, for example, which is a drug that inhibits ivermectin metabolism, we have done that in pigs and show that if a pig is given ketoconazole, which is a molecule that has similar effects to ritonavir on the cytochromes, and you feed mosquitoes on those pigs that have taken ivermectin alone or ketoconazole alone or both, then you match the concentrations of ivermectin versus ivermectin and ketoconazole, and what happens is that the mosquitoes feeding on the combined one die much faster because you're inhibiting the metabolism of ivermectin in the mosquito as well. In the pig, what happens is that you get much higher concentrations. So you want to be careful with drug-drug interactions. So we have our own trial. It's a small pilot in Spain. We have a very slow recruitment because we launched the trial and then there were no cases anymore in May. Now things are taking off. And let's see if we can finish the trial before the end of the year. This protocol is available online, fully available. If you want all forms, I'll give them all to you, including the statistical analysis plan. But then along came fraud. Fraud is this Surgisphere scandal. You may have heard about this Lancet retraction about hydroxychloroquine and how this huge paper showing 96,000 people. Well, it was based on what seems to be fake data. Well, the same authors had also meddled with ivermectin and created two preprints that show that ivermectin reduced 80% COVID mortality. Well, those preprints also suffer from the same issue of the Lancet ones, which is basically people believe they're fake. And here they are. This is the first version of the preprint showing this huge gap in survival in ivermectin treated people and non-ivermectin treated people. There were huge issues here. Numbers of patients didn't match. Then they changed for a second version. There's also huge issues here. These numbers don't match the text. And here again, 90% or 80% reduction in mortality. But the damage is done, and many countries have taken this up as national policy. Peru is taking it as national policy, Bolivia, some areas of Brazil now, and stuff like this is happening, right? You have a group that wants to save poor indigenous people, and they walk in and inject 5,000 people with that veterinary ivermectin for sheep. And then we can argue about that during the questions, right, if this is okay. So there's 32 active trials for ivermectin and COVID, either alone or in combinations. We'll see where we're going. I don't know. If it works, great. I hope it would work. My expert guess is that it might help as part of a combo, but that's just an expert guess. We need the data before we actually do any policy there. And I'm going to stop here. I talked for about 35 minutes.
Thank you, Carlos. Super fun. So guys, if you have questions, put them in the chat. I'm gonna start. I have a question. I didn't have the time to completely read the slide when you were talking about ivermectin and scabies, but then you had data for impetigo as well.
Yes.
Do we
to go back to that?
Yeah, sure. But I'm just curious about how ivermectin would work on a staph or streptococcus.
It's not because it kills the bacteria. It's because people are less itchy and they're scratching themselves less. So that leads to less self-infection.
Mm.
I don't think it's anything to do with an antibacterial effect. It has to do with an anti-itch effect and an anti-scabies effect that reduces people from scratching and infecting their own skin.
Okay, cool. Have you thought about looking at the impact of BOHEMIA on other arthropods? Because now there is evidence that ivermectin could affect, as you said, ticks, but also bed bugs. We know that bed bugs are quite a big problem here, and even if there isn't anything that is being done, when you go to the villages and you show them a picture of bed bugs, oh yes, we have this. So have you thought about this?
Yeah, so I haven't told you this yet, but we have a new PhD student, and she's going to be focusing entirely on NTDs in the context of BOHEMIA. But this is like the broad scope NTDs, so we're going to throw in there some bed bugs as well. We're going to focus on scabies, head lice, and tunga, but we also have considered bed bugs. It's trickier to measure, you may know.
We need to talk, Carlos.
I know.
Okay. Well, so there are a few questions being posted from Philip saying, what about efficiency of this treatment over 30 years old? Are various diseases resistant not evolving? I'm not sure I get you.
Yeah, so what he's asking is if there's ivermectin resistant onchocerca, for example, ivermectin resistant LF.
Years old, okay, it's my fault.
Yeah. So the existence of ivermectin resistant filaria has been something of huge debate. I think the official term, so what happens when you take ivermectin and you have onco is that these adult worms are producing microfilaria that are circulating in your body, and that they fix in your skin, they get into your eye, they stay in your blood. So what happens when you take ivermectin is you kill them all, this microfilaria, they disappear from your skin. And that is very quickly. So what's happening in some places in north of Ghana is that they are not cleared as fast. It's not that they are not cleared at all, it's that there's a delayed response in microfilaria clearance. It's not been named resistant yet because you can still use ivermectin. The good news is that moxidectin has now been approved for human use against onco, and it will probably follow soon against scabies because moxidectin rules against scabies much better than ivermectin. Against LF, there's not resistance, but we are killing them now with this combination. And on mosquitoes, that's a very, very, very complex question because mosquitoes seem to have differential susceptibility to ivermectin depending on their species, and that happens. For example, Aedes, you can basically not kill with ivermectin. It's impossible. You would need huge levels of ivermectin to do that. And Anopheles, there's differences between Anopheles species. So Anopheles gambius seems to be very susceptible, while Anopheles dirus or Anopheles aquasalis seem to be less susceptible. We don't really know what's happening there yet. And we think it's something species related, although the other thing you can ask me is, What about insecticide resistance? Because we know some of the mosquitoes develop resistance to pyrethroids by having metabolic resistance. They basically create more enzymes that clean or clear pesticides from their bodies. Could this happen to ivermectin again as well? Well, it could be. There is a theoretical risk. It's yet to be proven, because ivermectin shares some of these metabolic pathways with some pyrethroids.
Okay. Question are popping in. A question from Jack Sylvestre saying, would you recommend ivermectin as a first-line treatment for head lice infestation?
It's recommended for head lice in Brazil. It's part of the national therapeutic guidelines. You're probably aware of the French work on ivermectin and head lice. There's been a group there pushing for this for a long time. It's been approved as a shampoo. So now there's Sklees is called, and that's an ivermectin shampoo, and it works. I've used it in my children, both the commercial one and one that I homemade with ivermectin and shampoo at home. It's much cheaper, and it works great. Orally, I have seen it used. I gave it once to someone who was desperate, and it works. But then the risk-benefit analysis of a drug that is given orally, and it's still an oral drug, against an ectoparasite is not as good as a systemic parasite like onco. So I don't see this becoming a first option in the therapeutic armamentarium against head lice anytime soon, at least not orally, but it is in some areas, remarkably Brazil.
Another question from Franklin Aisi, I guess is in Ghana, is saying, how safe is ivermectin? Ivermectin is distributed through MDA generally once a year. For how long and how frequent can one administer ivermectin to stay within the safe levels?
Well, once a year, 200 micrograms kilo. In Kenya, in the Ivermal trial, they gave 300 micrograms kilo for 3 days in a row, and even 600 micrograms kilo 3 days in a row as well. And they had some side effects that were dose dependent. Interestingly, just as the ones seen in India when they gave, or Pakistan, when they gave 800 micrograms kilo single dose. So apparently this has to do with the Cmax that is reached at high concentrations, and but the side effects were mild, and they were basically visual disturbances that were self-limiting. Volunteers, healthy volunteers, have taken 2,000 micrograms kilo. That's 2 milligrams kilo. It's 10 times the normal dose, and they have no problems. So the therapeutic index is huge for ivermectin. It's a very safe drug, but also because of that, we're allowed to use it in MDA. So if people were to start using it at very high doses very frequently, and then there were some issues, it could hamper these mass drug administration programs, which are key to the eyesight of so many people. So very safe drug, up to 10 times the dose, fine. Watch out, because we could damage a lot of people.
Here's another question from Jacques saying, do you have an idea of the frequency of adverse events, particularly neurological, when treating lower hyperthalamic patients with ivermectin?
It's low. It's low. I think it's only seen in those above 15,000, and particularly those above 30,000 microfilaria per milliliter of LOA. I don't know the numbers off the top of my head, but there's not been thousands of cases. I think it's in the hundreds worldwide. The problem is that if you get encephalitis in one of these very rural areas, basically you have seen the number of respirators in some of these countries, right? So if you need a respirator for a couple of weeks, then your chances are not very good.
Yeah, because he's saying that they didn't treat scabies with ivermectin when he was in Cameroon because of the possibility of filariemia.
Of course. Cameroon is endemic for Loa, and in some areas of north of Cameroon have a highly endemic for Loa. So that means there's a risk of possibility of people having more than 30,000.
Oh, okay, so I have one last question, unless somebody is writing a question now. What happened to the people that were injected with ivermectin in Peru? Haven't followed up on that.
Well, the story was given by some journalists. In the journal article, they described that they didn't feel well. It's not very well described though, so it's difficult to assess. What is being reported is deep skin ulcers in those that are being injected with veterinary stuff. And there's even a few photos published online and in a Peruvian journal of internal medicine. It's also there. Celis is his last name, one of the doctors that have talked more about this. So, but I don't think the issue is the skin problems. I think the problem is that there's a huge risk of, instead of injecting 0.1 milliliter, you inject 1 milliliter. And then you have given not just 10 times the dose, but 10 times the dose subcutaneously, erratic absorption, and this also opens the door to all sorts of hell. Like now there's people getting injected with Adamacin in Peru. Adamacin is a derivative from ivermectin. It's never been used in humans, yet people are getting injected with it. So what happened? I don't know. So what happens always with indigenous neglected communities, right? People come, they do as they please, and then very few are accountable.
Okay, I think that was the last question. If anyone is writing something right now, please raise your hand so I can just wait a bit. Yep, no, I think that was the last one. Thank you so much, Carlos. It was super nice. And I'm going to text you on WhatsApp now.
Okay, we chat.
Okay, we chat. Okay, thank you, everyone.
Thanks, everyone.
Yeah.
Bye.
And I was wrong last time. I said that the next talk, this talk would be Sylvia. Of course, this is not Sylvia talking. Sylvia is talking next week, but malaria as well, but I don't want to reveal too much, but it's very cool. You should all join. Thank you so much, Carlos.
Bye.
Bye.